Hello, and thank you for joining my presentation today. I would like to talk to you about genomics at the Victorian Clinical Genetics Services, or VCGS, and how improvement in technology continues to allow us to improve the service we deliver to our patient. VCGS is a fully accredited clinical genetics service and is a not-for-profit subsidiary of the Murdoch Children's Research Institute in Melbourne, Australia. We are located at the beautiful Royal Children's Hospital in Melbourne. Like the little meerkats present here, we are always on the lookout for something new. Translation and research translation in particular, as a partner for MCI, is one of our core deliverables. In addition to our fully integrated clinical genetics and laboratory service, where we can draw on a large multidisciplinary team of scientists, geneticists, genetic counselors, bioinformaticians, and many others to deliver our work. VCGS is active across a vast range of genetic and genomic services, from preconception and prenatal and newborn screening all the way through to diagnostic testing throughout a patient's life, in particular delivering whole exome and whole genome sequencing. We were one of the first laboratories in Australia to be accredited in the space, and we continue to be one of the largest providers. Obviously, the evolution and revolution in genetics is driven by technology. In the past, in the very early days, which I was lucky enough not to have to be present for, genetic sequencing was done on gels with manual counting of bases and their associated positions. And the first revolution was really brought about by the emergence of automated Sanger sequencers. These were the machines to sequence the first human genome, but for delivering genomics at the scale we are doing today, these were also not feasible. It's only the emergence of next generation sequencing technology that now enables us to sequence dozens of human genomes a day. In comparison, it's really like the transport revolution. If your goal is to visit every country in the world and all you have is a horse, that's really not a feasible proposition. A car is theoretically possible, but still not realistic. We want planes and fighter jets to fulfill that task properly. But even with these changes in technologies that have now been well ingrained in the healthcare system, or in many healthcare systems, genomics is far from static. Technology continues to evolve and continues to pose new opportunities and new challenges. And some of these is what I would like to talk to you about today. In particular, the continuous improvement in the field of bioinformatics allows us to do more with existing data and more with data that we're going to create in the future. And really, it enables increasingly the analysis of variants that were previously intractable by genomic technology, such as CNVs or copy number variants, structural variants, and even repeat expansions and mitochondrial DNA analysis are now perfectly feasible. In addition, the emergence of multi-omics integration is going to continue to shape our field. Today I'm going to talk about an anecdotal example of how transcriptomics helps us to interpret genomic data. But even established technologies like whole exome sequencing continue to evolve with better chemistry, which improves coverage, uniformity, allows more automation, and allows us to push the boundary of what is possible in terms of scale and cost, even with tests that have been around for many years now. So I would like to start to talk about one of the areas that I find highly interesting, and that's the ability of genomic data and bioinformatics tools to create CNV calling in an increasingly reliable fashion. Here we can see an example of a rather complex CNV in a very difficult region of the genome, which is the CFH operon. And in this case, on whole genome data, we managed to resolve this family that had overlapping deletions in this area of different sizes. But even though this is an example of whole genome calling, we're increasingly becoming better at calling data of whole exome sequencing as well. And an in-house developed tool we use for that purpose is called CXGO, which is a visualization that you see here that utilizes the output of multiple CNV callers to deliver large and small copy number variants of the data. But this data can not only be used to operate in the kilobase to tens of kilobase range, even on a whole chromosome scale with different plotting methods, we can now detect whole chromosome events, as this example of what looks like a large deletion of large part of the X chromosome and turns out to be a ring X in this particular patient. CNV, the gap between cytogenetics and molecular genomics has now truly closed through the emergence of this technology. This does bring additional challenges with, when it comes to interpretation, because these fields traditionally operated quite separately, and these are the ones we now have to address to ensure that this information is consistently interpreted and also in context of the corresponding molecular data coming with the same cases. Genomic technology doesn't only allow us to analyze nuclear DNA, though. An increasingly common occurrence is the analysis of mitochondrial DNA from existing genomic data, again, in particular, whole genome data, but also from dedicated whole mitochondrial DNA sequencing assays, and even some whole exome sequencing assays have plenty of coverage over the mitochondrial genome to allow for reliable analysis. For mitochondrial, we generally get several hundred to several thousand-fold coverage, which is important because we have shown that we can, with high reliability, detect even low-level heteroplasmic variants in our patients. The sensitivity of some of our assays is as low as 3% heteroplasmy. But not only can we detect the point mutations in mitochondrial DNA, we're also able to call the copy number variants, where we use raw coverage, bin coverage, and split read plots to screen for breakpoints in the mitochondrial genome, which allows us to find common deletions, like in this case with Pearson syndrome, as well as other presentations, like this patient in panel C, where we have a multiple mitochondrial deletion syndrome, which in this case was triggered by a nuclear DNA variant that caused issues with the mitochondrial repair mechanism. The emergence of these whole mitochondrial DNA assays really allowed us to increase our diagnostic yield compared to the more targeted assays we had in the past, where we used different technologies to find either known pathogenic point mutations or separate assays to find mitochondrial deletions. And we managed to increase diagnostic yields in our patients by, depending on how we look at the data, by between 60 and over 100%, so almost a doubling in diagnostic yield. Especially if we compare our now single assay that manages to find deletions and point mutations to the separate assays. If we take into account likely or variants, good candidate variants, VUS variants that have a high potential of becoming likely pathogenic in the future, these are variants that are not detected by the traditional targeted assays. Our diagnostic yield potential more than doubles compared to what we have done only a few years ago. One of the areas that is now slowly starting to emerge and has traditionally been said cannot be done on this type of short-read genomic data is the detection of repeat expansions. And here I would like to give you an example of a tool developed by one of our collaborators, Andreas Hallmann, that is called Stripey, which utilizes short-read NGS data from exomes or genomes to make assessments on repeat expansions. The technology remains limited by the read length and the inset size that is available on the common short-read sequencers. However, even with those limitations, we can quite reliably call many repeat expansion diseases that are where the pathogenic expansion rate is within the range of these limitations. But even repeat expansions that go larger, that are falling slightly outside that range, are still tractable. All the way to the largest repeat expansions like a myotonic dystrophy, where while we cannot reliably ascertain the size of the alleles, we still get a strong indication that there is a potential expansion present. Now, I should say this is still technology in development. It's certainly not reliable yet and needs orthogonal validation quite extensively. But it has given us some really good indication in some cases. And one example, again, I would like to present here, which is a child that came in with features of hypotonia and remained negative after trio whole genome sequencing. We didn't find any causative variants. We ran the Stripey tool and we found what looked like an expansion in the myotonic dystrophy locus, where the tool in first pass thought that was over 50 reads, which is the pathogenic threshold. The issue here is with myotonic dystrophy to have a childhood onset, you really, or congenital onset, you really want expansions of over 1,000 repeats, which is obviously difficult to do. But with a bit of additional tweaking of the algorithms and manual analysis by Andreas, we managed to show that it's probably over 200 repeats. We're in the process of orthogonal validating the exact expansion size, but we're quite confident that this is the correct diagnosis in this case. Now with that, I would like to switch to the topic of multi-omics integration, and as mentioned, especially the area of transcriptomics, in particular an example of RNA sequencing. Transcriptomics is, in my opinion, highly valuable as a DNA genomics companion assay, which can be used to clarify candidate variant effects or identify new candidate genes. RNA, however, is compared to what we're doing with DNA, a very challenging sample. It's unstable. The library prep and the collection can be quite difficult. And it certainly takes a bit of extra work to get good data out of it. However, in our experience, we don't need the best quality data to actually get useful information out of RNA. And we've run a pilot where we extracted RNA directly from EDTA bloods rather than the more RNA-specific blood collection methods that are often used these days, and used batch sequencing on 8 cases as a pilot where we hadn't previously had a clear diagnosis. For those of you that don't commonly work with RNA, this is an IGV screenshot of an RNA sample of the exist locus, where we can nicely see the 2 types of data we can obtain from RNA. One is splice information, where we get spanning of reads from coding region to coding region of the exon. And the other is expression information, where in this female case, the RNA is expressed from the exist locus, from the second X chromosome as expected, where the 2 male cases in this case don't have exist expression because there is no second X chromosome where X inactivation would be required. The case I would like to present is a case where we had a high clinical suspicion of a diagnostic or pathogenic variants in the RMRP gene, which was pretty much a priori the only gene that could explain this phenotype. The problem is that the gene is non-coding. So despite the fact it's recessive and non-coding, despite the fact that we found a good candidate insertion variant in the proband that was inherited from the mother, because the gene is non-coding, the normal frame shift and NMD interpretation rules do not apply. So we couldn't assess the impact of this particular variant. In addition to that, the second variant that we would have expected, which is a common promoter deletion, wasn't present, and what looked like reasonably common point mutations and a bit of noise in the data was certainly not interpretable. We progressed to trio RNA sequencing as part of our pilot and got a number of very informative results. First of all, we could show that the expression in the father was downregulated by 4-fold, and he's the one that we would have expected to have the suspected promoter deletion, while the mother did not have any significant change in expression. The proband, on the other hand, had over 30-fold downregulation. So what happened here? Turns out on further investigation in the data, we got on the DNA fooled in the first pass by our aligners, and the insertion in the promoter that we would have expected was indeed present and also showed up as alternative transcription start site in both the proband in the father. So the variant that we initially thought wasn't there, based on the RNA data, we did some further digging in the DNA data and could ultimately identify that. So that explains the downregulation in the father and to some degree the downregulation in the proband. However, interestingly, the insertion variant that was inherited from the mother, while clearly present in the proband, is not visible in the RNA sequencing data from the mother, only in 1 out of several hundred reads. So that indicated to us that the transcript with the insertion was unstable, and that upon a bit of extra literature research, transcript instability is a common mechanism in this particular gene caused by these insertions. And while our particular variant had not been previously published, with the RNA data, we could then prove the transcript instability in the mother, which explains the combinatorial downregulation of expression in the proband, and the case was ultimately solved. We learned a few lessons from this pilot, most importantly that RNA data doesn't have to be perfect to yield useful information. A inverted commas quick and dirty method still can be very informative. And in this particular case, we actually got quite lucky because 4 out of our 8 pilot cases yielded some form of valuable information. 2, the case I presented, and an additional case actually managed to be diagnosed with the help of the RNA data. Plus, we managed to confirm one splice variant that we had previously found, but also refute an additional splice variant that was found as a candidate in a different case. Trio sequencing in RNA absolutely adds value, as we've seen in the case I presented. We could not have cracked without the information from the parents. But there is still a lot of work to be done and a lot of challenges to be overcome to make RNA sequencing data really as a proper diagnostic test. We would like to see it as a concurrent standard test to genomic testing, either genome or exome sequencing. Lastly, I would like to cover improvements in established technology. As I mentioned, just because the technology has been established in routine clinical care doesn't mean that no improvements can't be had. And we learned that lesson ourselves a bit more than a year ago when we evaluated a new chemistry from Twist, the new Twist exome chemistry. And we ended up with an assay that ended up being both higher quality and more relevant, but also cheaper than what we've had before. So why was the assay more relevant? It's because we could work very closely with Twist on a new design or an improved design, which was worked up between them and us to optimize clinical yield in a diagnostic setting, both by boosting coverage in Mendelian gene regions, but also by specifically designing in known pathogenic variants in non-coding regions, which we would not have captured by our previous exome methodology. But we also showed that we managed to achieve better coverage with less sequencing. And this is what I would like to show you next. The Twist assay compared to our previous assay, which we show the coverage here at 100x sequencing, performs equivalent at a percentage of bases covered greater than 10x metrics to our previous assay, even if we only sequence at 80x target coverage, and arguably even at 50 and 40x target coverage only. When we look at the percentage of bases covered over 20x, the assay actually performs better, even with 20% less coverage. So overall, that was a strong indication that we can actually get away with less sequencing and have the same high quality data. When we looked a bit further, we actually found that there is the number of gaps in the data is also substantially reduced. We define a gap as any base that is less than 10x coverage, and the percentage of bases with the cumulative size of regions with less than 10x coverage dropped dramatically in even lower coverage, 80 and 50x coverage samples from Twist. So in summary, by investing in new chemistry and design and validation, we ended up with a test that has much higher quality data, is more clinically relevant, and actually allowed us, in addition to offer a better clinical exome test, allowed us to to consolidate a number of our previous targeted smaller panels into a single test, because it turns out that the exome chemistry ended up cheaper than the array of different targeted capture panels that we were previously offering. So at VCGS, we are now offering only exome and genome-based testing, and our target panels are all in silico on a twist exome backbone. Again, with continuous improvement in technology, there remain key challenges to overcome, in particular, the continuous requirements to improve and validate your chemistry can be a challenge. However, we now see that as part of our routine service because the benefits we get from that are so many that for us now continuous improvement is part of routine service delivery. Nonetheless, we have to continuously keep an eye that the technologies we're testing are also scalable and cost efficient so we can use them in an accredited diagnostic setting. But the key to our approach is really the continuous integration between clinic and research, and we work very close with our collaborative partners to translate the emerging learnings from the research setting into our clinical setting and then feed back to inform the next round of research. One last pitch I would like to make is for Panel App Australia, which is a tool that has been developed by my collaborators Donitza Stark and Oliver, as well as Ellie and Augusto, in collaboration with Genomics England and Australian Genomics, which is really the local version of Panel App, where we use crowdsourcing for gene curation, and we are now adding new knowledge for new genes at a rate of roughly 25 to 30 genes a month. So if you're interested in gene disease association, I'll ask you to come visit our Australian version of Panel App, and please feel free to contribute. With that, I would like to thank my many collaborators and the teams that they are leading, as well as Australian Genomics, Melbourne Genomics, and the Genomics Health Future Missions in Australia that has been partially funding some of the work presented today. I'd like to thank you for your attention, and I will be available for questions afterwards.